Hello friends, I am Dr. U S Desai working as an assistant professor at PES Modern College of Pharmacy, Nigadi, Pune. So today we will see the negative staining technique. So following are the requirements for carrying out the negative staining technique. First is the bacterial suspension. Second is glass slide. So you'll be requiring two glass slides. Nichrome wire loop, nigrosin or eosine stain and microscope. So now we will see the principle involved in negative staining. Now as we had discussed in last video of monochrome staining, the stains which we used in monochrome staining were basic stains, but here in negative staining we will be using acidic stains, for example eosine, nigrosin, which carries negative charge on it. And as you all know, the bacterial cell wall is made up of peptidoglycans. So it contains proteins, and protein contain amino acids. The amino acid contains both carboxylic group and amino group. But the carboxylic groups are oriented outwards. So when these carboxylic groups undergo ionization, they carry negative charge, that is COO minus. So, as the acidic stains are also negative in charge and your cell surface is also negative in charge, the stain is not retained by the bacterial cell wall and it gets deposited around the surface, resulting in appearance of bacterial cell as colorless against dark background. So now we will see the procedure of negative staining technique. So first you have to clean the glass slide. As you all know, the glass slide has to be cleaned first by water in order to remove the dirt or dust contamination, and later the slide has to be cleaned by any disinfectant like ethanol, which will prevent the contamination of microbes, if any, present on the glass slide surface. The next step is preparation of smear. Now here what we'll be doing is we'll be taking the one loop full of bacterial suspension, and you will deposit that bacterial suspension at the end of the glass slide, not at the center of the glass slide as we did for monochrome staining. We'll be putting one loop full of the bacterial suspension at one end of the glass slide, and then we'll be adding 2 to 3 drops of the nigrosin stain at the same end of the glass slide. Then with the help of another glass slide, we will mix the two things, that is the dye as well as your bacterial suspension, loop full of bacterial suspension, and then by holding the another slide at 45-degree angle, you will pull that slide across the another glass slide and prepare a smear. Then you will allow that smear to air dry. Heat fixing is not required in negative staining because you are not going to wash the stain with water after drying, since the stain is going to be deposited in the background and there is no chance of excess stain. So here heat fixing is not required, air drying is sufficient. So once the surface has been air dried or the smear has been air dried, you will put a drop of oil, that is either cedarwood oil or liquid paraffin, for proper magnification and observe it under 40x objective lens in microscope. So once you observe the slide under 40x objective lens under microscope, the image appeared will be like this. Here you can see the background has been colored with the stain, that is either dark violet or almost dark purple in color, and all those colorless spots indicates the bacterial cells. So here you can see the cells are appeared as colorless and the whole background has been colored where the stain has been deposited. So by using this kind of technique, you can observe the bacteria in the form of unstained cells. Thank you.